Come on. Recording. Sharing my screen, if this will show up. All right, you should be seeing my screen now. It's not switching. There it is. So today is August 17th. We're approaching the end of the class. This is the last week before finals week. We will be having a lesson on Tuesday of finals week, and then the final will be Thursday, August 26th. Any questions about that? The final will be comprehensive. It will have heavy coverage of the material that we covered since the quiz. The last quiz covered chapter 8. So that means chapter 13, chapter 14, and lab 9. No, I take that back. Lab 9 is on quiz 5. So just chapter 13 and chapter 14, about 50% of the final will be over chapter 13 and chapter 14. I suppose a little bit will be on the unknown report. So you should know how to do the case studies. Meaning if I give you the information, you can figure out which bacteria is causing the infection. There won't be many questions like that though, because those questions take time and your final will be mostly multiple choice, and that'll be a timed exam. There will be a few essay type questions. Let me look that up. Be certain about that. Stopping the share because I'm going to look into the final and see if there is yeah, there is essay questions, final A. It looks like there's only 3 questions. Come on. One of them will be where I give you some information and you have to figure out what bacteria it is. So it'll be like your unknown notes. All right, any questions? If not, we get out of here. And share my screen again. Please note that I did some more grading. I don't remember what I did now. I graded a number of the labs. Let me stop sharing the screen. I'll go to the grade book and then it'll be easy for me to see. So a number of the labs have to be corrected by Saturday. And I don't remember if that was this Saturday or last Saturday. So let me look. I know no, that's worksheet, sorry. The last lab I graded, yeah, that's it, lab 8. It's going to be due this Saturday. Any questions about that? Let me see if there's anything else. Yeah, lab 7 was due this Saturday, so. I did start regrading the older labs. That didn't go back to the grade book, just a minute, yeah. I know lab 0, lab 1, and I think lab 2 have been regraded. So if you're curious about that, you can take a look at it. I started lab 3, but I didn't get very far in lab 3. Where is that? Yeah, only lab 0, lab 1, and lab 2 have been regraded. All right, any question about any of that? I'll work on getting lab 9 done this week. And I'll work on getting lab 3 and lab 4 at least regraded this week. And maybe if I'm lucky, I'll get lab 5 regraded as well. All right, any questions about any of that? If not, let me go ahead and show the screen again. I think we never discussed this, but we're going to work on chapter 13. And we shouldn't finish it, but we should get close to it. Today in the lab, there is no lab. I'll be logging in at 6:30 to 6:45 to answer any questions. And I'll stay forever long it takes to finish the questions. But at 6:45, if I'm done and nobody's asking me any further questions, I'll log out. You should be working on your unknown work. If you're all done, you can start writing up your unknown report. If you are still working on your unknown work, get that done. The last day that I will be sending out results for the unknown project is Tuesday, August 24th, because Wednesday the report is due. On Tuesday, don't send me any test requests unless that test can be done within the day, meaning there are a few tests which have no incubation time, like the catalase test. If you request a test on Tuesday and it has 2 days incubation period, which is most of the tests, you will not get the results in time for your unknown project. So Tuesday will be the last day I'll be sending out results for the unknown project. All right, any questions about the unknown project? All right, let's, I'll keep this up. Let's get started on the lesson. Is this the last slide I talked about in this class? Viral structure, and I mentioned that there are 3 general viral shapes: the helical viruses, the polyhedral viruses, and then the complex viruses. Is this the last slide we talked about?
Yeah.
Oh, thank you. I just wanted a confirmation. All right, let me go in there and delete that. Come on. So T4 virus, I already mentioned, is the virus that has been most studied in the world. And the real reason for that is that T4 infects E. coli a cell which is very commonly found in the microbiology labs. So they chose T4 to study. T4 is in the class of viruses called T even, and it includes T2, T4, and T6. They all have this shape. They have the capsid head, which includes, I don't know, the capsid that has the DNA molecule in it. And then the capsid tail, which is this portion here. Many of the bacteria viruses have a tail, and the tail is a special structure for the virus to get through the cell wall and then infect the bacteria cell. Animal viruses may not have a tail because it's much easier to infect an animal cell. We only have a cell membrane, and it's just easier for the virus to infect the cell. Like I said, the tail is a special structure for infecting the cell, and many bacteria viruses have a tail. We call viruses that infect bacteria bacteriophages, or you could say bacteriophage. I'll try not to use the shortened name. Sometimes they call that a phage or a phage. I'll always use the term bacteriophage to make it clear to you that I'm talking about a virus that can infect bacteria cells. We will be mainly talking about viruses that infect bacteria cells or animal cells, because these are the viruses that are the most studied. And obviously we want to talk about animal viruses because those are the ones that infect humans and cause diseases. We will barely talk about plant viruses, and we don't talk about fungal viruses at all. There are viruses for the archaea bacteria, but we're not going to talk about them. Am I missing anyone? Oh, there are viruses for the protists as well. And we're not really going to talk much about those. I think at the very end, I'll discuss viruses that attack some of the protists. It's only one slide, not a big deal. Any questions about any of this? Bacteriophage T4 has the capsid, which has the DNA inside the head portion, and then the tail that includes the sheath, includes the base plate and the tail fibers. The only part of the virus, at least T4, that attaches to its host cell, which is E. coli, are this portion and that portion and that portion and that portion. The bottom of the tail fibers are the portion of T4 that attaches to its host cell, which is E. coli. The rest of the virus does not attach to E. coli. They are important for infecting the cell, and I mentioned that the tail is important for infecting the cell. The base plate gets rid of the cell wall, and then the tail actually contracts, and that puts pressure on the head, which has the DNA, and with that pressure that injects the DNA in the head into the bacteria cell. And we have a little video talking about that. Whenever viral infection occurs, at least of an animal, the host immune response will respond to it. Infection with a virus stimulates the host to produce antibodies. And we're not going to talk much about that, but it also can produce T cells. But antibodies recognize and bind to viral surface proteins. The portion of the viral surface protein that the antibodies bind to, we call antigens. And we discussed that a little bit earlier. Antigens tend to be the outermost molecules of a virus or an invading host cell. And on this virus, it would be the spikes, the outermost molecules on the virus. For this virus, it would be the protein coat that makes the capsid, would be the antigens. What the antibodies bind to and the host recognizes as foreign and responds to. Some viral proteins can change or mutate frequently, and that means there can be different viral strains, and therefore they can infect the same individual more than once. A very good example of that is influenza. It changes its surface proteins fairly quickly. And that's why when you get an influenza vaccine, the vaccine is good only about every 3 years. And after 3 years, we say the vaccine is giving you so low of protection, you should go and get another influenza vaccine. Now, some viruses don't change very often. Like, for example, the polio vaccine was made in the 1950s, both of them, and they are still used today as the vaccine against polio. So obviously the polio vaccine is not changing, not mutating. And so the vaccine, I don't know, how long is that? That's over 70 years. Of course, depends which year in the 50s they were made, but you're getting the point. And I think I'm doing 1950, and it's closer to 1956 and 58, something like that. But you get the point. Some viruses can change frequently, like the influenza virus, and some viruses change slowly. Most viruses change slowly, and that's why the vaccines are still good that were made in the 50s, like the vaccine to yellow fever, polio, the measles vaccine, I think was made in the 1960s. And these viruses are all still good, the vaccines are still good. COVID-19 is one that changes in between influenza and something like polio. And probably you will need a booster shot about every year for this one. Well, that's not like, that's actually closer to influenza, sorry. They're not certain yet because the vaccines haven't been out that long, but they're talking about it's probably a good idea to get a booster 8 months to a year after you got the initial vaccine to COVID-19. Of course, this virus is lethal, and so that's why they're suggesting you get the vaccine booster 8 months to a year. And they haven't picked the time yet because, like I said, they're still studying that. If you don't know, Delta variant is a mutant, and that's the virus that's currently infecting the United States with COVID-19. And I'll talk more about viruses, or at least COVID-19, later in the lesson. Any question about any of this part? All right, so viruses are taxonomized the same as cells. However, viruses are really only grouped into families. You can group them into, let me see if I can remember this. Sun, moon, over. You can group them into orders. Very few viruses have a grouping larger than an order. And that's mainly because they're small. They evolve pretty quickly to their host specimen, and viruses, I guess, just evolve quickly. And so we're not really sure about their evolutionary history because they're small. They don't have that much DNA or RNA, and it changes. And so we're not real sure about their evolutionary history. And that's why we don't group them into very large groups. There is one exception, and that is we do group viruses into the family, or I should say the domain that they infect, or the kingdom they infect, like animal viruses. And we know that it is an animal virus because those are the cells it infects. When we sequence the DNA, it's not so clear that that virus infects an animal cell, just because there's not enough DNA to base it on. But we do group them into the kingdom that they infect. We also group viruses based on the type of nucleic acid they have, whether it's DNA or RNA, the replication strategy the virus has. We'll talk more about that. The morphology of the virus, whether it's a helical virus or a polyhedral virus. We do group the viruses, all viruses, into their families. And the family name of a virus ends in viridae, like the herpes family would be the herpes viridae. We then group the viruses into their genus. All genus names end in virus. So the herpes genus, I don't know which one that would be because that's the family, but it would be the herpes virus. So all genus names end in virus. We then specifically state the viral species. The species is a group of viruses sharing the same genetic information, and they share the same ecological niche, like HIV-1. It's all similar when we sequence the virus, and then they all infect human cells, specifically human T helper cells. You'll be pleased to know that when we group viruses, we give them a common English name, like the herpes virus or the polio virus. We do not have Latin names for viruses. And it just happens that the early virologists were all English speaking, and so they started naming them with English names. And then the later, languages that started studying the viruses just kept the English names because that's what was already there. Like the French-speaking virologists, they started using the English language names. Does that make you happy? You don't need to learn any Latin names for the viruses. They all take common English names, like the polio virus. Nobody's going to comment on that. All right.
Yes.
I thought you'd be happy with that. Let me put that over there. We then further group viruses into a subspecies. Subspecies are designated by a number. Subspecies are very important for a virus. Now, for the Delta variant of COVID-19 and the Alpha variant of COVID-19, they're not all that different, although the Delta variant is more communicable and a little more serious, meaning the patients are more severe than the alpha variant. But those are subspecies. You also have subspecies which are very different, like for the polio virus, which I assume that's number 1. The subspecies number 1 causes the disease polio. A subspecies of the polio virus we use as the Salk vaccine to prevent getting the disease polio, meaning the subspecies is used as a vaccine to prevent people from getting the disease polio. So hopefully that shows you how important it is among viruses to discuss the subspecies, because one subspecies causes the disease polio. The other one we use as a vaccine to prevent getting the disease polio. Any questions about any of that? All right, so the subspecies can be very different. They do infect the same host, by the way, and they have the same genetic information, but they can be very different on how they cause disease. And I suppose when I'm talking about the alpha variant and the COVID-19 Delta variant, they are different. And that is the COVID-19 Delta causes a more severe disease than the alpha. And the Delta is also much more communicable. Or I could say contagious. Have we talked about those terms yet? That might be the next lesson. All right. An example of viral taxonomy for the family, we call it the herpes viridae. All families end in viridae. The genus would be the herpes simplex virus. The common name, which is not being used very much by the medical community and the public at large, is the human herpes virus or human herpes simplex. And the human herpes simplex is the common name which is being kept. The human herpes virus is not being taken by the medical community. The subspecies would be human herpes virus or human herpes simplex virus type 1. All right, any question about viral taxonomy? If not, let's talk a little bit about how you can grow bacteriophages in the lab. Remember, a bacteriophage is a virus that grows in a bacteria cell. What you do is you just grow up the bacteria and then inoculate the media with viruses. And then the viruses that infect that one bacteria will then grow in the bacteria. I've actually done this. One thing you can do is plate the plate with bacteria so that bacteria will grow in a lawn. That means the bacteria will grow everywhere. And then you put on a solution containing the virus. And then you incubate that plate, and wherever the bacteria is, it'll look whitish or off-white from the bacteria growing there. And where the viruses are, they will grow in the bacteria and infect that bacteria cell, replicate, and then go on to infecting neighboring cells, and then lyse those cells. And then you'll see a plaque, and wherever there's a plaque, that means there was a virus there that infected the first bacteria cell and went on to infect neighboring cells, and that causes the plaque on the plate. Any question about any of that? Now for the difference in plaque sizes, this one is actually, you can see it, 3 plaques that fuse together. But that one there looks like it's only one, and that one there is only one. And this one here is smaller, and that one there is really small. Why are they smaller? Well, this one here that's big, probably the virus, which is what we call the plaque, touched the bacteria cell initially and then started replicating initially. So when the virus replicated, it lysed that cell and then went out and infected other cells. Whereas this cell here, or this virus here, and this virus here, or there, let's go there, it's easier to see. What happened was the virus initially wasn't touching the bacteria. So it's on the plate, trying to show that here. It's on the plate here. And it had to wait until a bacteria nearby replicated, and then touched the virus. And then the virus could then replicate and start lysing the bacteria cells around it. So this cell, this virus probably didn't replicate initially. And so that's why it's a smaller plaque than this here. The plaque is just where all of the bacteria have been lysed. And this is the color of the agar without having bacteria. And then this is the color of the agar with the bacteria growing on it. Any questions about any of that? Viral multiplication happens when the viral genome hijacks its host cell. The virus only has a few genes in it, and by itself, the virus cannot replicate. It needs to hijack its host cell and then make the host cell replicate the virus. So following infection, the virus takes over the host cell's metabolic machinery. An easy way for the virus to do that is to degrade the host cell's DNA. And then your genome is calling the shots for what's going on in the cell. The virus uses the host ribosomes, the host tRNA, the host amino acids, the host nucleotides like ATP, and some of the host enzymes to replicate the virus. The basic mechanism for viral multiplication is similar in almost all cells that viruses infect. Viral multiplication in bacteriophages happens this way. The virus infects the host cell, which in this case is a bacteria because we're talking about bacteriophages. And then it causes that cell to replicate the virus. Bacteriophages have 2 life cycles, which are 2 separate mechanisms for replication of the virus. In the lytic life cycle, the host cell is killed and lysed, why it's called the lytic life cycle. The other life cycle that bacteriophages can engage in is the lysogenic life cycle. And in the lysogenic life cycle, the virus does not kill the host cell. As long as the virus stays in the lysogenic life cycle, the host cell remains alive. It is infected with the virus, but the virus is living in symbiosis with the bacteria cell. And that is why we call it the lysogenic life cycle. Any questions about these 2 life cycles? Different viruses follow different life cycles. So if the virus is a lytic virus, it will only engage in the lytic life cycle. If a virus is a lysogenic virus, it will engage in the lysogenic life cycle, but it may also switch to the lytic life cycle. And we'll talk more about that when we're talking about the lysogenic life cycle. When a virus needs to replicate, what does it need? What components does it need to make in order for the bacteriophage to multiply? What's the virus need to do if it wants to replicate itself? Come on, somebody guess. I'm not going to move forward until there's at least one guess.
It needs
Get
a
inside
bacteria
the
cell.
cell.
Say again, I didn't hear that.
It needs a bacteria cell.
Yeah, it needs a bacteria cell, but what viral components does the virus need before the virus can replicate itself, meaning one go into two?
Proteins.
Yeah, it'll need to make protein, specifically the capsid proteins of the protein code of the virus. It'll also need to replicate the viral genome. And some viruses, that's all the virus needs to replicate. It will replicate if it's a DNA virus, it will replicate the DNA and make new viral DNA. And then it will transcribe that DNA. Blow this up a little. To make viral messenger RNA. And then that will be translated to make the viral capsid proteins. And then the capsid proteins have to get together with the viral genome to make the new progeny virions. So that's the way viruses replicate. Any question about any of that? And this doesn't have to be a double-stranded DNA genome virus. The virus can have a different genome. You just have to replicate that genome. I'm only showing it for double-stranded DNA because it's easiest to discuss. All right, let me see if my movie's going to work here. For the viral multiplication, it can happen in two life cycles. The first we're going to talk about is the lytic life cycle, which goes through 5 stages. Let me go to my thumb drive. I've still got it here because I'm not using my headphone, which I should be using. Let me see if I can play that movie. I'm afraid it uses Flash, so I might not be able to play it. I'm not seeing it there. It might not open 2 of these. Let me change that. Because it has the same name. What slide was that? 19. Ah, I got the wrong one. Sorry, let me go back.
First, the phage particles stick to the surface of the bacterial cell. They bind to specific sites.
Well, that's not very good. Well, this is taking a little while. Well, let me go through talking about it and we'll see if that'll play. There are 5 stages for the lytic life cycle. First, there's attachment. This is when the bacteriophage attaches to its host cell. This happens with the tail fibers. I don't know if you can see this right here. There's the tail fibers right there. Next comes the stage penetration. Phage lysozyme in the tail opens up the cell wall. So there will be a hole in the cell wall. The lysozyme digests the peptidoglycan. And then the tail sheath contracts, and that forces the DNA in the head into the bacteria cell. Next comes biosynthesis. That is production in the host cell of the phage DNA, if it's a DNA virus, and the phage proteins, the capsid proteins. That's followed by maturation. The phage particles, meaning the phage protein, gets together with the DNA. It makes the newly assembled viruses. And then lastly, what the virus does in the lytic life cycle is it releases the enzyme lysozyme, and that will dissolve the cell wall. And then water will move into the host cell, and the cytoplasm will expand until the cytoplasm bursts. And when the cytoplasm bursts, that will release the newly replicated bacteriophages. Any question about any of that? Oh gosh, I don't know the thing here, so let's just try it again.
First, the phage.
No, it's not gonna run. All right, sorry about that. You have to listen to me suffering. You can't get the movie there. Although there's another movie. Right here. Oh, shoot. That's why I opened up the second one. This file, oh, that doesn't look good. It's a different movie though.
In a way, a virus is a parasite. It attacks a host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall. and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle.
Okay, any questions? Oh, maybe I should use this one. Let's go back to where we were. So initially in the lytic life cycle, the virus will attach to its host cell, as I specified. Only the ends right here of the tail fibers are what attaches to its host cell. The binding is specific, meaning there are specific molecules on the end of the tail fibers, which bind to specific molecules on its host cell. If the host cell lacks those molecules, the virus will not attach to it. Like, would be a good example. Staphylococcus epidermidis is another bacteria, and T4 will not attach to Staphylococcus epidermidis because it's lacking the receptor molecules on the host cell. Any question about any of that? All right. After attachment, there's penetration. The sheath secretes in the area of the base plate the enzyme lysozyme, and that puts a hole in the cell wall. The sheath then contracts. Was that shown in the movie? I don't remember. It might not have been shown in this movie. The virus contracts, and that puts pressure on the head. And remember, the DNA molecule is in the head. And with that pressure, that literally shoots the genome, it doesn't have to be DNA, but we'll talk about it with DNA, from the head into the host cell. And so the DNA gets injected into the host cell, much as when you put pressure on the needle, you put the contents of the needle into a person. So the virus is acting much as a needle, and that is this sheath contracts, that's not contracted and contracted, and that pushes the contents of the head into the cell. And that's how the virus injects its genome into the host cell. Then biosynthesis happens. The viral genome takes control of the cell, and an easy way to do that is it can encode an enzyme which degrades the host DNA. And then your genome is calling the shots in the cell. Anyways, the genome gets replicated, and the genome gets transcribed into proteins. The capsid proteins come together, and in this case, the tail proteins have to be made as well. And then the tail proteins and the head proteins come together, and the genome gets put inside the head. And that we call biosynthesis or maturation. And then lastly, when the new virions are made, the virus genome that has infected the cell encodes the enzyme lysozyme, which lyses the cell wall of the bacteria cell. And then water just moves in, and the cytoplasm expands until the cytoplasm breaks the cell membrane. And that will release the newly replicated viruses, which we call virions. Any question about any of that? There are two terms you should know in the lytic life cycle. Burst time is the time it takes for a virus meaning a virus species, to infect its host cell and the time till the virus bursts or breaks the cell. This averages between 20 and 40 minutes, dependent on the viral species we're talking about and the temperature. Any question about that? The other term you should know is the burst size. That's the number of newly released, newly replicated virions that come out of the infected cell. And that's usually between 50 and 200, but I'm not going to ask you about how many. You should just know the terms. All right, any questions about the lytic life cycle? All right, bacteriophages can also replicate following the lysogenic life cycle. In the lysogenic life cycle, the virus, if it follows the lysogenic life cycle, does not lyse its host cell. The two live together in symbiosis. Once a lysogenic virus first infects its host cell, it has to decide immediately upon what it wants to do. And it can do one of two things. It can enter the lytic life cycle, in which case, where do I have that shown? Oh, it's come here. In which case the virus replicates itself, makes copies of itself, they assemble, and then it lyses the cell, releasing the newly replicated viruses, which then go out and infect neighboring bacteria cells. Any question about that? The other thing the lysogenic cell can do is it can enter the lysogenic life cycle. And what it does is it will stay in the lysogenic life cycle, keeping the host cell alive. What happens with the lysogenic life cycle is it makes a repressor protein. What the repressor protein does, it shuts down the genes for the lytic life cycle. And then the cell remains in the lysogenic life cycle as long as the repressor protein is active. So a lysogenic virus, when it first infects its cell, it has to decide, do I want to enter the lytic life cycle or do I want to enter the lysogenic life cycle? If it enters the lysogenic life cycle, a couple of things happen that are important. First, the host cell remains alive. The viral genome incorporates itself into the genome of the host, meaning if it's a DNA virus, its DNA merges with the host DNA, and then the two become one chromosome. We call the viral DNA in the host chromosome, we call it the prophage. So the purple, or I should say the reddish purple, is the viral DNA, and now it's a prophage, but it is part of the chromosome for this host cell. Now, the only time the virus replicates in the lysogenic life cycle is when the host cell replicates its DNA. When this DNA replicates, the prophage will get replicated as well. And then the cell will divide, and then one copy of the chromosome goes to this side of the cell, and the other copy goes to this side of the cell, and then the cell splits in two by, I'm drawing a blank there. What the heck is that called? Whatever. This can go on through many cell divisions where the host chromosome replicates and then the cell divides into two. Every so often, for reasons that are still not clear, the lysogenic virus can leave the lysogenic life cycle. And if it does that, the viral genome excises itself from the host genome, and then it becomes a separate genome. And then the virus enters the lytic life cycle, going through the destructive lytic life cycle, destroying the cell, releasing the new viruses. Now, this is a lysogenic virus. And so once it infects this cell, once again, it has to decide, will it enter the lytic life cycle or will it enter the lysogenic life cycle? All right, any questions about any of that? Why does the virus choose one or the other? We don't really know. And like I said, they've been studying why the virus leaves the lysogenic life cycle for over 50 years. I think it's over 70 years now. And they still don't have a good understanding about why the virus leaves the lysogenic life cycle. Just sometimes it does, and then it enters the lytic life cycle. Now, with that stated, they did find one thing that can cause the virus to leave the lysogenic life cycle. And that is if you damage the DNA of this cell, the virus will leave the lysogenic life cycle and enter the lytic life cycle. The reason being is the virus probably knows the DNA has been damaged, and with a number of damage to the DNA, meaning you have a number of them, that can be lethal to the cell. And so the virus then will enter the lytic life cycle to replicate before this cell dies. All right, any questions about the lysogenic life cycle in general? If not, let's talk about it in greater detail. If the lysogenic virus decides to enter the lysogenic life cycle, its phage DNA, which is circular, will combine with the host DNA, it integrates, and they will become one chromosome. This we call the prophage. And then it will replicate whenever the cell replicates. Okay, eventually it will come out, and then if it does, it will enter the lytic life cycle. We've already talked about the repressor protein. As long as this repressor protein of the lysogenic virus is active, it shuts down the lytic genes, the genes responsible for running the lytic life cycle. All right, any questions about any of that? If not, let's talk about some consequences of the lysogenic life cycle. Lysogeny is the name of the life cycle, but you call it the lysogenic life cycle. If you have a cell which is infected with a virus, which is following the lysogenic life cycle, that cell will be immune to infection by the same bacteriophage, meaning the same virus, same species of virus cannot infect that cell more than once. And so it will remain in the lysogenic life cycle. The other consequence for the lysogenic cell is that this cell here had its genome there, which was a dark, is that a green color? Not sure of the color there. let's say dark green. And after the cell gets infected, this cell now has new DNA, and that DNA will be incorporated into the host cell chromosome. Well, this new DNA can bring a new gene to this cell. And then this cell can express a new phenotype. And that's a consequence of lysogeny. It has new DNA. That DNA can code for something, and that can change the properties of this cell. A perfect example is the Corynebacteria diphtherium. This bacteria only produces the Corynebacterium diphtherium toxin when the bacteria has a lysogenic virus infecting the cell, meaning the lysogenic virus encodes the gene for the Corynebacterium diphtherium toxin. So yes, how sick you will be dependent on the bacteria infecting you. And if the bacteria is infected with a virus, a lysogenic virus, at least in the case of Corynebacterium diphtherium, you will be sicker if there is a lysogenic virus in the bacteria. If not, you have Corynebacterium diphtherium growing in you, but it will not produce the toxin. Any question about any of that? I think we talked about toxins once before. A toxin is a substance that is toxic to us or harmful to us. And toxins generally kill some of our cells. Of course, if you have too much of the toxin, it will kill you as well. All right, any questions about the consequences of lysogeny or the lytic life cycle in general? If not, let's move on to talking about animal viruses. We talked enough about bacteriophages. Let's now move on to talking about animal viruses. These are viruses that infect animal cells, and they follow the basic pattern for phage multiplication that we discussed earlier for bacteriophages. There are several differences, like differences in attachment. The virus has to attach to the cell membrane because animal cells have no cell wall. It does follow the same response, and that is the virus will have a surface molecule which will bind to a receptor on our cell before the cell can be infected. And if you have a cell lacking that molecule, the virus will not bind to that cell. And I used to know an example of that. I'm not bringing it to mind. The penetration can be different. We'll actually talk about that. The penetration can be by endocytosis or by fusion if it's an envelope virus. But the point is the virus can penetrate the cell, and it's a little different because it's an animal cell. And then you have encoding of the virus. That's pretty similar. Biosynthesis, that's pretty similar as a bacteriophage. Maturation, that also is similar to the bacteriophages. The release of the animal viruses can be different. You can have release of the animal virus by rupture of the cell, and this we call a lytic animal virus. So there are some of those. An example is polio. The polio virus is a lytic virus, or it follows the lytic life cycle. Any question about any of that? But the virus doesn't have to follow the lytic life cycle. It can follow another life cycle. Sure, what we're going to talk about is the release can be different. And instead of the lytic life cycle, we can have the budding life cycle. And this is where the virus literally replicates in its host cell and then buds out of the host cell. Usually the virus does not kill the host cell if it is in the budding life cycle. I'm sort of getting ahead of myself. If the budding life cycle kills a host cell, it's usually the immune system that is responsible for killing that host cell. The immune system kills that host cell because it finds that it's infected with a virus. All right, any question about any of that? The multiplication of animal viruses follows the basic pattern of phage multiplication or bacteriophage multiplication, but there are several differences. The main difference I want to talk about here is that in bacteria, the virus has to replicate in the cytoplasm because there's no other portion to the bacteria cell. That's not always the case in animal cells. And the animal virus can replicate in the nucleus, or the animal virus could replicate in the cytoplasm. It depends on what species you have. Some viruses replicate in the nucleus, some viruses, animal viruses, replicate in the cytoplasm. Any question about any of that? We'll talk about a rule of thumb for determining where the animal virus is replicated, either the nucleus or the cytoplasm. Attachment can differ. As I mentioned, animal viruses, when they attach to their host cell, they can penetrate by endocytosis or by fusion of the lipid bilayer. Let's talk about endocytosis. I should mention that the older version of the textbook had an error, and it called it the virus entered the cell by pinocytosis. It's not pinocytosis, it's endocytosis, or I'm trying to remember what the 3 types of endocytosis are and which one it follows. Anyways, the virus, when it attaches to the cell membrane, it then causes the cell membrane to start invaginating where the virus is. And that invagination will grow until the virus is literally inside the invagination. The invagination then breaks off, and then this membrane, it's a lipid bilayer, will then attach to this lipid bilayer, and then the virus will be brought into the cell by a lysosome or a phagosome. Any question about any of that? Oh, and the cell membrane, when it breaks right here, it will reassemble right there. So you have cell membrane attached to cell membrane right there. And then the envelope here will attach. I don't know if you can see that. So it'll break off and then attach. And that's how we get the phagosome. surrounded by a lipid envelope. Any question about any of that? Attachment and penetration can also happen by fusion. If the virus has a lipid outer coat around the protein coat, the lipid bilayer causing the envelope is a bilayer, and it can actually fuse with the bilayer of the cell membrane. And so let's put a virus like here in the cell membrane. They come and attach, and then they fuse. And then that opens up. I don't know if you can see that. There we go. And that will allow the virus to come into the cytoplasm. And then this portion of the envelope will bind to the cell membrane over here. And now the cell membrane is literally, let's use it for this cell. The cell membrane is literally coming up like this. And then instead of going across, it's going to go over the virus. And what it's going to be doing is that's going to be the envelope. And then it'll come down here and attach to the cell membrane again. And that's how viruses, an animal virus, can infect an animal cell by either endocytosis or by attachment to the cell membrane. And where does that go? Fusion. Drawing a blank as to what that was called. All right, I think we'll stop here and I will continue here next time. Any questions? All right, you don't need to show up for the lab, but I will be there from 6:30 to 6:45 to answer questions. All right, I'll see you on Thursday.